This video will introduce the structure of eukaryotic chromosomes during cell division. As cells prepare to divide in M phase of the cell cycle, DNA must be dramatically compacted so that it can be efficiently moved to daughter cells. So, prior to M phase, our chromosomes undergo replication in the S phase, followed by additional compaction. During this process, nearly all of our DNA is in the form of heterochromatin and is thus not transcriptionally active. Let's begin with a short review of how our DNA is condensed in our cells when they are not dividing. Our DNA double helix is wrapped around histone octamers to form nucleosomes that look like beads on a string. This shortens our DNA by about sevenfold. Next, interactions among nucleosomes due to the histone H1 protein further shortens our DNA by another sevenfold into the 30 nanometer fiber. Loop domains are then formed through the interactions of the 30 nanometer fiber with two different proteins. This step organizes our DNA into areas that are transcriptionally active, those in the loop domains, and those that are not, which are those closer to the protein binding sites. Step C here shows how our chromosomes exist in our cells when they are not dividing. In preparation for division, though, these loop domains are further condensed and folded up on one another in preparation for forming our final, highly condensed metaphase chromosome. There's still some debate as to which proteins do what in the final condensing to the metaphase chromosome. Some evidence points to a combination of histone and SMC proteins, but at present, this is still in the plausible hypothesis stage, so stay tuned for the final answer. We do, however, know that there are two protein complexes that are crucial in the formation of the metaphase chromosome. The first is condensin, which is important in condensing the loop domains at the very beginning of mitosis. The second is called cohesin, which is crucial in sister chromatid alignment from S phase through anaphase of mitosis in the cell cycle. Each of these two important proteins contain SMC proteins, which are a class of proteins crucial to the structural maintenance of chromosomes. Condensin is actually found in the cytoplasm, and during interphase, condensin 2 enters the nucleus and begins the process of folding up the loop domains. However, the final condensation into the metaphase chromosome doesn't actually occur until the end of prophase in mitosis, when the nuclear envelope dissolves and condensin 1 can bind to the DNA and complete the final compaction of the chromosomes into what we recognize as the metaphase chromosome dyad. Our other important protein, cohesin, is important in the cohesion of sister chromatids. Beginning at the end of S phase, this protein keeps our sister chromatids aligned and attached to one another. These proteins remain bound as condensin binds and condenses our chromosomes during prophase. Towards the middle of prophase, the cohesin proteins release from the chromosome everywhere but the centromere. It is this protein that holds our sister chromatids together as the spindle fibers migrate them to the central axis of the cell during metaphase. Finally, during anaphase, these final cohesin molecules release, allowing the sister chromatids to separate and migrate to opposite poles of the cell.